welcome back to the lecture series in cell culture technology. So, in the last lecture we talked about the mechanical dissociation of the hippocampal tissue, and this whole mechanical dissociation process is uniform. I am just taking up one simple example, and one has to be slightly careful while you are doing mechanical dissociation, while you are kind of offering a shear force to the piece of tissue to dissociate it into single cell suspension. You ensure that you are not picking up air bubbles. Those air bubbles damage the tissue big time. So, you will always have to do it very gently. So, there are some thumb rules out here in mechanical dissociation. You have to be very gentle and fairly smooth process. It has to be done with utmost care. The other method which especially for this particular thing I will not recommend, because and I will give you a few references, cite few references, you will see. What you can do is, say for example, in this case you have this is extracellular matrix. So, you follow something called enzymatic dissociation. We will come later where you really needed enzymatic dissociation. Enzymatic dissociation essentially means, say for example, these extracellular extracellular matrix has say collagen. Okay. We have already talked about collagen, you remember. So, what I will be using to break this collagen? What I will do, I will use an enzyme which will break the collagen. I will use collagenase, ASE. Whenever it means it is an enzyme. So, I use collagenase enzyme at a limited concentration. You have to be very careful what is the concentration you are using, or I can use another protease which will break these bonds of collagen. I can use papain. What is papain? This is one enzyme which is derived from papaya. We all eat this as a vegetable as well as a fruit, but this papaya, which is Carica papaya, and you must have seen many people say if you are having a stomach upset or something, or like you should take some papaya. What it does, it essentially the way papain works is that, say for example, and this will be very important in the phase 2 of it, you will realize why I am kind of. So, say for example, this is the tissue and these are the cellular elements in the red dots, right. Now, what papain does is, let us represent papain molecule as green, it kind of infiltrate into these zones. Okay. And the job it does is weaken the adhering forces between the cells. This is what it does, and in that process followed by this treatment, if you do offer a mechanical shearing or mechanical dissociation, what you obtain is again the same thing what we talked about here, a single cell suspension. But this single cell suspension in this case goes through two processes. You first did a papain processing followed by a mechanical dissociation. But when we talk about the fetal tissue to isolate hippocampus from mother mice or rats, like for the fetus, you really do not need to go through this whole rigor or go through this whole, you really can do it mechanical dissociation. So, now what you are having, once you have mechanically dissociated it, now you have this in a tube. So, for example, this is the tube where you have collected. So, you have a single cell suspension in a medium. So, again, as of now we have not talked about medium, just assume that you have a particular composition of medium where these cells will grow, or a buffered solution, okay, supplemented by different growth factors. So, this is your medium, okay, and here in red dots I am showing the single cell suspensions. Here they are sitting. Okay. Now, what you do, you take these and beforehand, okay, having said this, beforehand you have to prepare your cover slips or substrate. So, in the last class we talked to you about how to prepare substrate. You have to prepare the cover slips, you have to coat them with the substrate of your choice. So, in this case for hippocampal neuron, one of the favorite choice is poly D lysine. I will come to other substrates where it is even much more preferred than poly D lysine, but at this stage just keep that in mind. So, you have here is your cover slip, and here you put medium in which they will grow. Okay. Before putting medium, I have said this, before putting medium you take a small part of this cell suspension, and of course, I missed out on one point is these are poly D lysine coated cover slips or glass. Okay. So, now on this what you do, you take this suspension, small amount of suspension first, and you try to plate the cells like this. You just put them there, they will find out with little bit of a medium in that, but try to plate them and without adding any further medium for next 30 to 40 minutes, and then after 30 to 40 minutes add more medium. This is where you are submerging it, but do not go very high. I told you, just good enough, because so that they can derive the oxygen from the environment easily. Okay. Add more medium, and of course, again put it back in the CO2 incubator. We will come back to the CO2 incubator and the buffering system. So, this is the zone. You remember I told you that there has to be everything defined. So, this is your how we do the buffering. We will come to this very soon, and here is your, put it back in CO2 incubator for buffering, and here also well you left it in the incubator. Okay. And now you monitor the growth. So, what I haven't told you as of now is this other part, composition. What is that medium? This I haven't told you. CO2 incubator fundamental concept in terms of the buffering I haven't told you, but I have told you about the substrate and how you are isolating the cell. So, I have defined two things. If you look at carefully, I have defined two things. This is all defined and this part is defined. Now, once you have plated the cells and you monitor them under the microscope, so this is what you are going to see. These single cell suspension, these cells will regenerate and they will form a random network depending on how close or how at a distance you are plating them. So, typically there is certain cell density of plating what you have to do. So, this is another thing which I haven't mentioned here, cell density. Mostly for these such small neurons are typically at the rate of, I would say, anything between 50 to 150 cells per millimeter square, typically, depending on, because there are two things you have to realize here about the cell density, and this has to be defined very clearly. Either you follow 50 or you follow 150 or you follow anything in between, say 75 or 100. I personally has always typically worked with 100 cells per millimeter square, unless otherwise there is a demand for a very low density culture. So, when we go for 50, we talk about a very low density, when we talk about 150, these are high density cultures. Again, there are people who may disagree, they can go a little higher also, but I personally, after working with hippocampal neuron for more than a decade, I will not recommend that. So, if you go very low density, the problem is for the cells to, so cells, they grow in colonies. And when we grow in a colony, we have interaction with our neighbors, in the information transfer and all those kind of stuff which happens, which is strengthened. Say for example, your neighbor tells you, there is this problem happening, so be careful, or water will not come on this day. So, so it's kind of mutually helping each other in a colony. Cells are exactly like that. They are colonies. They are colonies where they continuously crosstalk with each other. And if you sparsely seed them, so one is here, another is another 1 kilometer away, another, so that interaction is going to be missing, and some of these interactions are survival concept or survival therapies for them. They survive because they crosstalk, they interact. So, that could be one of the problem one could face when we talk about a very low density culture, and will come. There are situations where you have to do a very low density culture. We will talk about patterning and all that kind of stuff. We will be coming soon to this. But if you do a very high density culture, you can, so the problem is that then there will be a resource crunch. It is just like you put too many people in a confined location and now you have resources, that is not an issue, but how the resource will reach to the cells which are kind of covered by other cells, and it creates an anaerobic situation, and there are a series of problems which, because the way the metabolism will function, it will be functioning at a tremendous pace, and you do not want to get into that kind of situation, right. So, you always have to optimize what rate you are going to follow, but then having said this, whatever density you follow once, you have to keep on following that, you cannot change it. So, how this is being done is, once you isolate the cell, then you do use a hemocytometer to quantify the harvest. In this case, cell. So, once you have quantified the harvest, so you know how many cells per ml you are having here per ml of medium, per ml of medium. Now, if you know this many cells are there per ml of medium and you want certain specific number of cells to be plated in a certain area, what you have to do, say for example, I say I want, let us take a practical situation. So, I say, so there are say 10,000 cells per ml. Okay. Now, I want to plate, so I say I want to plate in a cover slip whatever size, I am not defining that at this stage. I say I want to plate, say for example, okay, let us take a number now. Okay. So, I say I have a cover slip of say 10 millimeter by 10 millimeter. Okay. So, that makes it 100 millimeter square. Accept it? Now, I said if I have 100 millimeter square, I want a density of say 100 cells per millimeter square. If I want 100 cells per millimeter square, then I will be needing 100 millimeter square multiplied by 100 cells per millimeter square. So, your millimeter square cancel. So, what you are having cells. So, what you are having is 10,000 cells. So, you have 1 ml. Now, you have to spread this 1 ml on these cover on one single cover slip in such a way that it uniformly spreads in the beginning. So, the cells now will spread all over the place in a uniform way. So, you are taking 1 ml, and of course, you may have a stock of like 10 ml. So, you can place 10 cover slips in that situation. So, depending on what will be your density, you have to do the back calculation here very carefully, and you have to be very meticulously careful. You cannot afford to have in one culture I did 100 cells, the other culture I did 200 cells, and the third culture I was somewhere in between. Every time the data what you will be deriving from it will go haywire. So, please be very careful. These are things which are not being told, but as graduate students, as an independent PI, you have to be cautious, you have to be very careful. If you are a PI, then you have to ensure that your lab staff follows this, or if you are a graduate student, that is even much more important, because this is your experiment which everybody wants to repeat at some place in the world, and they should not come across, come on, I mean this time this is the effect I see, the next time I see another effect. These small errors can cause very dearly. Okay. So, now once I have told you about how you, so this hemocytometer calculation, this you can really pull out any manual where they do the blood analysis, will figure out how much you have to put and how much cells you are seeing. So, this is okay. Fair enough. So, you plated the cells. Now, once you have plated the cells, so this is called the cell plating. So, if you remember, I told you that go back to the master chart, what all you have to be careful. So, it is here, cell plating rules, remember, I told you that this you have to be very careful, how much cells you are plating. So, when we talk about defined condition, these are those small parameters which will come very handy. We talked about the isolation rules, then we talked about, let me add one more in your master slide, cell count, and followed by now cell plating. So, making sense how small details are going to influence the results you are going to interpret. Okay. Now, coming back. Okay. Here I was. So, you have done the cell plating. Now, what will you observe? Within, if it is a hippocampal culture, within 24 hours, you will start seeing these cells will be sending out processes, and within 2-3 days, you will see these cells are growing very neatly. But they will have a very random connectivity all over the place. Who is forming synapse with which cell cannot be dictated. Now, I told you this industry wants to do an Alzheimer's testing. So, it puts the drug, which is say represented by X drug or Y drug or Z drug, into this medium. And one more thing, whenever you are going this primary culture, the first medium change what they are recommended is what happens after, generally it is recommended after 24 to 48 hours, you should change half the medium, just half the medium. You warm the medium in a bath and just replace half the medium, and followed by that at every fourth, depending on what is the density of culture, third or fourth day, change 3/4 medium. Some people completely change the medium, which I not fully recommend for a reason, because the cells secrete lot of factors which are helpful for their survival. So, if you remove everything out of it, so that the cells are devoid, they have to again redo the whole exercise. So, I would say 3/4 of the medium you change every 4th day likewise. Okay. So, you have these drugs now at your disposal. So, what I will do, I will close in here. In the next lecture, we will talk about how these drugs are going to act and how the company is going to interpret, and what all next gen problems which will come, what they have to define. Okay. Thank you.